Hello friends. So first of all, I would like to thank you all for your immense support. So far, I have uploaded certain GCEL related videos. But from today onwards, I thought of uploading GCEL biology related videos covering the main aspects of your curriculum, of your subject. So let's start from today this process. So to start with, I thought of touching the basic unit of us humans. So what do you think it is? What is the basic, maybe structural and functional unit of us humans? You can pause the video for a while and yeah, just think about it. Yes, it is the cell. As we all know, the basic structural and functional unit of us humans and of course living organisms is the cell. So in multicellular organisms like humans, the cell, kind of more cells combine together and they form tissue, and there is, when tissues are kind of formed together, they form the organ and then the organ system, and then the organism is finally formed, that is we humans. But there are some unicellular organisms as well. For example, most of the prokaryotes are unicellular. But we all know that this cell is a characteristic of living organisms. So there are several characteristics of living organisms, such as they have an order and organization, but that don't fit to everything because certain organisms are unicellular, but they show metabolism, there is this hereditary and evolution. So there are this stuff. So if you want to recap, you can just go to your resource book or your book which you have to refer, go and check your first unit, your GCEL biology first unit. So it's they have listed the characteristics of these organisms. So the cell is very important. So we are today going to have a look at the cell. So what is there in this cell? What are the internal organelles we can find in these cells? So usually we have an idea that the cell is actually a 3D structure which is kind of spherical. Here you can see a spherical cell. But is that true? So just think about it. Do you think that all our cells, don't think about different organisms, just take humans, do you think that all the cells in us humans are spherical, or we can find different type of cells which are in variety of shapes? Could you think about it? Just, if you want, just pause the video and think about it. Alright, let's have a look at it. Red blood cells. We all know about red blood cells. Actually, it makes a major part of our blood. So to speak, about 45% of our blood is made up of these red blood cells. So here you can see the shape is different. When you look at this outer appearance, you can see the biconcavity. Here you can see this side is concave and this side is also concave. So it's a biconcave disc where you have a thickened rim. Outer rim, it's thickened. Here you can see the thickened rim, and here you can see the central pallor. It's actually flat. You can see the thickness is reduced. So this is also a cell which is found in us humans. But this is not that usual cell we talk about. So here you can see the usual cell which has all these organelles and a spherical shape. It has a spherical structure and these organelles. But we all know that red blood cells lack organelles. They do not have organelles it is an adaptation to carry oxygen, mainly oxygen throughout our body. So what else? What is this? It looks like a beautiful drawing, but it's not. It's actually a nerve cell. We can call it as a neuron. So here you can see this part is known as cell body. And here you can find many organelles packed in this cell body. But here we have this axon and the synapses or synapses and the dendrites. So this structure is totally different. Actually, this is the longest cell we can find in our human body. So I just want you all to understand that when we talk about cells, there are different type of cells which look in different shapes. So they got these shapes because it's a functional adaptation. As I said, for red blood cell, it is a functional adaptation because it has to be biconcave, because you all know they travel through our capillaries, which are very tiny vessels. So they should be able to, these are actually not rigid structures. They can actually fold and they can go through these capillaries. So that's why we find so many cells in different shape, in different structure. So with that note, now let's take a look at the classical cell. So this is the normal cell usually we talk about. So let's have a look at the outer border of the cell, which is known as the plasma membrane or the cell membrane, and then we have these organelles. So let's have a look at all these structures which make up a cell. Maybe we do not find all these organelles in a cell, but some might be in a cell, some might not be. But when we talk as a whole, these organelles can be found in different cells. So plasma membrane, this is actually not a organelle, but this surrounds, actually this is the outer border of our cell. So fluid mosaic model of cell membrane, we all know that Singer and Nicholson, they actually proposed this model. So what is this fluid and what is this mosaic? So phospholipids are this fluid. We call phospholipids as fluid because we can see this is actually a cell membrane structure. Most of our cell membrane is made up of phospholipid. So here you can see most abundant type of lipid in plasma membrane is actually the phospholipids. So it's like a sea fluid, it's like a sea. And in between we can see this mosaic, that is these proteins you can find here. These proteins are like mosaic. So in the sea, it's like an iceberg. You can find these proteins. So it's easy for you to keep this in mind. So the fluid is phospholipid, and here and there you find this mosaic, which is known as protein, which is the protein actually. So phospholipids, talking about phospholipids, here you can find the phospholipid. The cell membrane or the plasma membrane is made up of phospholipid bilayer. There are two layers of phospholipids. Here you can see this blue part and here the yellow part. Actually they are arranged in opposite directions. The interior of this plasma membrane is made up of this yellow part of both the layers. So this yellow part is the tail of the phospholipid molecule. And then we have this blue color part, which is known as the head of the phospholipid molecule. They are facing the exterior. So if we take that this part is the inside of the cell, and if this is outside of the cell, we all know that most of this inside and outside is made up of water. So outside we find interstitial fluid, inside we have intracellular fluid, ICF, and outside we have interstitial fluid, we might call it as ISF. So this is water. And amphipathic molecules, why do we call these amphipathic molecules, these phospholipids? Because they have this water liking part and the part which doesn't like water. So this is the hydrophilic head and hydrophobic tail. So hydrophilic head is facing the water part because they love water, so they are facing the water part, the aqueous solution. But the tail, they do not like water, so they are arranged in the interior of the plasma membrane. So now you can understand why they are called amphipathic molecules, because they have hydrophilic head and hydrophobic tail, and why they are arranged in this manner. So first, talking about plasma membrane, mainly we talk about phospholipids, which is the fluid in the Singer-Nicholson model. Then we have protein, that is the mosaic. So So there are two types of proteins. One is integral proteins. Integral proteins, here you can see two types of integral proteins, and then we have peripheral proteins. Integral proteins could be transmembrane proteins or monotropic proteins. So transmembrane protein actually spans the cell membrane. They are fully embedded in this cell membrane. And integral proteins, they might be fixed into one leaflet of the phospholipid bilayer. If this is one leaflet and the other layer is another leaflet, this might be in one leaflet. That means it might be in this inner leaflet or in this outer leaflet. So these are also integral proteins, but they do not span the cell membrane, the whole cell membrane. Then we have peripheral proteins. Peripheral proteins, actually they are lying on this plasma membrane, outside or inside. So here we can see peripheral proteins. They do not get into this phospholipid bilayer. So there are two types of proteins, integral and peripheral. And then we can find few cholesterol molecules that is not depicted in this image. So few cholesterol molecules. So how do they look like? How will they fit? So here you can see if this is one phospholipid molecule and then we have another phospholipid molecule. In between two molecules, we might find a cholesterol molecule. So why do we have this cholesterol molecule? Actually what they do is, when the temperature is high, these phospholipid molecules, they are known, we call them as fluid because they have this movement, they can move. So when the temperature is high, they show certain movements that they can move. So to limit that movement, the cholesterol molecule is inserted in between two phospholipid molecules so that their movement is kind of limited. If not, they'll be moving like this. So it will be like a collapse. So the cholesterol molecule is inserted. But when the temperature is low, this cholesterol molecule will not be there. They will leave, because when the temperature is low, the phospholipid molecules are also kind of rigid. They do not have much of a motion. So when the cholesterol molecule is also there, it might kind of solidify this plasma membrane. So that is the function of the cholesterol. But in the normal body temperature, the phospholipid molecules show kind of a movement. So the cholesterol molecules are here and there to limit that movement. So that is about few cholesterol molecules. So now let's move on to the next slide. Functions. What are the functions of this plasma membrane? So it surrounds the cytoplasm of living cell, and they form a barrier in between intracellular and extracellular environment. So they are separating both the environments, because in intracellular environment we have different composition of ions and maybe molecules, but outside it's different because they are needed to be different to maintain a living cell, in a normal range it should be. Then selective permeability. We all know that cell membrane is highly selective. It has highly selective permeability because it cannot let all the molecules to get in. So they have these carrier molecules, they have pumps, so that they selectively, ion channels, they have channels, so they selectively let ions or molecules to get in or outside of the cell. Proteins embedded in the plasma membrane. So as I said, there are these proteins. So let's have a look at these proteins' functions. What do they do? They are involved in cell recognition, especially in the external side of the membrane, which is facing the interstitial fluid. Not the internal side, but the external side. We have these proteins. They actually are helpful in identifying the cell. So other cells can identify this cell to maybe to change certain signals and everything. So they are involved in cell recognition. Then we have certain receptor molecules which can kind of identify these hormones and initiate certain functions inside our cell. So there is this is our cell membrane, and there is this receptor, and this receptor identifies certain hormones. Maybe we all know that hormones or neurotransmitters, they come and signal our cells to initiate certain function. Then we have carriers, ion channels and pumps. All these are actually transporters. They are useful in transporting several ions inside and outside the cell. Then there are this cytoskeleton. We know microfilaments, then microtubules, intermediate filaments. These are the cytoskeletal fibers. So these cytoskeletal fibers can bind with these peripheral proteins maybe, so that they are useful in maintaining the cell shape. And enzymes. Outside the membrane, we might find certain peripheral proteins, and these might act as enzymes, so that for example in our gut, in our intestine, we have this peripheral protein which acts as digestive enzyme, so that when, for example, if maltose enters the intestine, then this enzyme can convert it into glucose and then absorb it. So these are the functions of the cell membrane. Nucleus, a very important organelle we can find in our cell, because it is controlling our functions and it's storing our genetic material, genetic information. So there is this nuclear envelope. It has two parts. One is the outer membrane, and then we have the inner membrane. Outer membrane is this, and then we have this inner membrane. Then nuclear lamina, this light pink color line, this is the nuclear lamina. It is actually surrounding the inner membrane. And then in the outer membrane we can find nuclear pores, so that certain molecules can enter and exit via this nuclear pores. Nuclear matrix, it is the internal matrix which is inside this nuclear lamina. We can find this nuclear matrix. In this matrix we find nucleolus, which is darkly stained, this nucleolus, and these filament like molecules are actually chromatin. Chromatin is actually loose chromosomes. The chromosomes are actually densely packed. Chromatins are known as chromosomes. So chromatin is also chromatin and chromosome both are made up of DNA and protein. So if here we have a protein molecule. The protein molecule is surrounded by this DNA, and when they are loosely attached we call it chromatin, and when they are packed together, so when it's like loosely coiled we call it chromatin, but when they are packed together it is known as chromosome. So this is what we find in nucleus. And what is its functions? So they control all the cellular activities, as I said, because it has our DNA, so obviously it can produce certain proteins and control our cellular activities. And then it synthesizes DNA so that it produces new nuclei, and it helps in cell division. So we all know in the cell division, before the cell division begins, we have this interphase, and there in S phase specifically, the DNA replicates, so that when, for example in mitosis, when the cells are divided, the genetic material is also equally divided. They synthesize rRNA, mRNA, tRNA. These are three types of RNA. So they are useful in protein synthesis. So these are synthesized, and of course they store and transport genetic information in the form of DNA. So in the DNA we have our genetic information stored. Actually in the nitrogen bases, in the sequence of that nitrogen bases. Ribosomes. Ribosomes, we have to talk about ribosomes because if you can remember this image, these yellow color tiny parts, these are actually the ribosomes. They are found on the nuclear outer membrane and also in this endoplasmic reticulum. Actually this is rough endoplasmic reticulum because it has ribosomes. So ribosomes, there are two subunits. One is small subunit and then the larger subunit. So here it's the small subunit and then we have a large subunit. So something like this. So it's made up of rRNA, which is synthesized in the nucleolus of the nucleus of the cell, and it has protein. So that is how this ribosome is made. There are two types, 70S and 80S. Both can be found in our human cells. 70S is actually found in mitochondria. The matrix of the mitochondria has this 70S ribosome. And also the prokaryotes, the stroma of the chloroplasts also have this 70S ribosome. So if you have any confusion in remembering 70S or 80S, mitochondria is actually a part of our cell. We have a big cell and inside it, a part of it is mitochondria. So it has this small ribosome, which is 70S. And when we take the whole cell, it's large. So it has 80S ribosomes. So 80S ribosomes are found only in eukaryotes. You should remember it because prokaryotes have 70S ribosomes. Well, so these 80S ribosomes can be free ribosomes or bound ribosomes. So free ribosomes are freely available in our cytoplasm, and bound ribosomes, as I said, are found in this rough endoplasmic reticulum. You can see in the outer surface and in this nuclear membrane also sometimes we find this ribosome. And this is actually, here you can see these tiny parts. These are actually, these tiny dots, these are actually the free ribosomes which are found in the cytoplasm. So the function is actually the protein synthesis, as I mentioned earlier. So this ribosomal subunit, the ribosomes, both small and large subunit make up the ribosome, and then mRNA, it carries the message, that is the genetic information from the DNA, and it comes and it gets into these two subunits. In between, when I drew it, you can find that there's a space. If this is the small subunit and this is the large subunit, in between here mRNA, this is an mRNA strand, it comes, and then this information can be coded and tRNA carries the specific amino acid, and that's how the protein is synthesized. Maybe that's not necessary now, so just that you can understand what's happening. So protein synthesis, we need these ribosomes. Then endoplasmic reticulum. There are two types of endoplasmic reticulum, rough endoplasmic reticulum and smooth endoplasmic reticulum. Earlier we saw that the ribosomes are attached to a certain endoplasmic reticulum, and that is the rough endoplasmic reticulum. Consists of flattened sacs and ribosomes bound to surface. So you can see flattened sacs. These are flattened sacs, and they have ribosomes bound to their outer surface. So proteins are synthesized by ribosomes and then they move into the lumen of the endoplasmic reticulum. So here the proteins are synthesized and they move into this lumen. So it's a sac, you can understand, it's a sac. So if you can take a bag in your home, take a shopping bag and look at it. So inside the outer, you have this polythene or something, the bag, then inside it's a space. So same here. So this is also a sac. So inside there is a space. That space is actually the lumen. So the function is, as I said, the protein is synthesized by these ribosomes which are attached to this rough endoplasmic reticulum. So they transport this protein synthesized by ribosomes. They get this the protein inside their lumen and then they transport it via their transport vesicles. So one function, they transport protein synthesized by ribosomes and then they produce transport vesicles to transport it. They transport it to the Golgi apparatus. So there are certain changes are made. Glycosylation, like carbohydrates are added to the protein synthesized. So these are two functions. Apart from these, they synthesize glycoproteins And also they facilitate the growth of their own membrane by adding phospholipids, proteins and carbohydrates. Therefore, they are called as membrane factory. So these are the functions of rough endoplasmic reticulum, and you should remember this. This is actually in your resource book. You can refer it for further clarification. Then we have smooth endoplasmic reticulum. These are actually network of tubular sacs without ribosomes. They do not have ribosomes, but membrane-bound enzymes are present. So the functions are here. They actually synthesize lipids, they metabolize carbohydrates, and they store calcium ions. Actually, the smooth endoplasmic reticulum, when they are found in muscle cells, especially in the cardiac and the skeletal muscles, they are known as sarcoplasmic reticulum. There they store these calcium ions. So storing calcium ions is also one of their functions. Then they produce, of course, they also produce vesicles for transportation, and they are involved in detoxification. So these are the function of smooth endoplasmic reticulum. So you should remember these as well. Golgi apparatus. So Golgi apparatus, we can move to our very first slide. So here we can see the Golgi apparatus. So these are also flattened sacs. So these flattened parts are known as cisternae. These are the cisternae. Each part of this is known as cisternae. And this part, the outer part, is known as the cis phase, and the inner part is known as the trans phase. So there are two phases. The cis phase is located near endoplasmic reticulum to receive vesicles from the endoplasmic reticulum. Trans phase gives rise to secretory vesicles. So inside we have trans phase, outside we have, so inside like this, we have trans phase. If it's like this, outside we get, this is the cis phase. This is the cis phase. The outside we have cis phase. So they have abundant secretory cells. Their function, they collect, package and distribute materials. As I said, vesicles from rough endoplasmic reticulum containing the proteins and everything, they come here. So they are collecting, and inside they package, and then here they create their own vesicles and they transport. Manufacturing cellulose and non-cellulose cell wall components such as pectin. This is actually a function in plant cells. And also they produce lysosomes. These vesicles, one of these vesicles are known as lysosomes because they contain these digestive enzymes. Next we have lysosomes. As we all know, lysosomes are formed from what? Golgi apparatus. So they are single membrane bounded vesicles contributing to digestive activity. As I said, they have digestive enzymes, so they can digest carbohydrates, proteins, lipids and nucleic acids. So they digest food particles received by phagocytosis. There are two types of endocytosis, that is engulfing the particles from the external environment into the internal environment, that is inside the cell. So if the solid particles, large solid particles like bacteria and some large particles are, if they are engulfed, it is known as phagocytosis. And if slightly if small molecules with liquid, like liquid particles, if liquid particles are engulfed, it is known as pinocytosis. So there are two types of endocytosis. So endocytosis is actually engulfing the particles from our external fluid, which is the extracellular fluid known as interstitial fluid. So they digest food particles received by phagocytosis. So how do they digest? They have this enzyme. So when the phagocytosed particles get in, the lysosomes kind of they both fuse, and the enzymes in the lysosomes help catalyze breakdown of these particles. transport residue material out of cell by exocytosis, and finally when these particles are digested, the remains are then exocytosed. Digest worn out organelles. So in the same manner, inside the cell also we have some dead organelles, so they can also be digested, and if there are any important nutritions, they will be absorbed within the cell, and the remains will be again exocytosed. Endocytosis is getting the particles inside, and sending the residue or any particle outside is known as exocytosis. Autolysis causing cell death. So they can also cause cell death by kind of, they destroy themselves also when they are kind of matured, or when they are dead, when they are matured, they do this autolysis, so they cause cell death. So these are the functions. Especially we should remember that they have this digestive enzyme, so they kind of digest everything. They digest organelles, they digest themselves, they digest food particles which are entering the cell, which are going to enter the cell via phagocytosis, and then after those are digested, the remains will be exocytosed. So this is the function of lysosomes. Peroxisomes. These are also single membrane bounded vesicles. Lysosomes are also single membrane bounded vesicles. These are also vesicles with oxidizing enzymes. These have digestive enzymes, lysosomes. These have oxidizing enzymes. So they are present in both plants and animals. So they kind of catalyze the breakdown of H2O2. These enzymes can break down H2O2. Hydrogen peroxide. It is also a peroxide. So detoxification of peroxides. This is a main function. Then photorespiration, which is usually we can find this in plants. So this is a function in plant. When the peroxisomes are found in plant cells, they are helpful in photorespiration as well, not in human body. So there are some specialized peroxisomes called glyoxysomes. These are only found in plant cells. They are found in actually fat storing tissues in plants. They convert the fatty acids. These are found in fat storing tissues. So in fat storing tissues we might find fatty acids. They convert fatty acids into sugar. So this is their function. Mitochondria. Mitochondria is also an important organelle we can find in cell. So talking about its structure, you should be able to draw this structure. So kind of when you are free at home, try to draw this structure. They have two membranes. They have one outer membrane and then inner membrane. Earlier we saw that glyoxysome, peroxisome, then lysosomes, these are single membrane bounded vesicles. But these have two membranes. The outer membrane, which is very smooth, but the inner membrane are folded inside. You can see here. These are folded inside here. And this inner folded, this part is known as crista or cristae. And then inside there is this matrix. So there we find this matrix. Also inside in this matrix we can find ribosomes, 70S ribosomes and DNA. But this DNA is circular. That is a difference from the DNA that is found in the nucleus and mitochondria. The mitochondrial DNA is circular, but the DNA we find in nucleus is actually the DNA, which is the double helix ring. They have two strands and they are intertwined kind of. They form this double helix shape. So that is the difference between mitochondrial and a nuclear DNA. So keep that in mind as well. Cytoskeleton. We earlier also discussed that the plasma membrane proteins, the peripheral protein which is lying in the inner surface of the plasma membrane, it is actually connecting with some cytoskeletal fibers because they are helping in maintaining the shape of the cell. The cytoskeleton usually maintains the shape of the cell. So there are specific microtubules, microfilaments and intermediate filaments. There are three types of fibers in cytoskeleton. So microtubules are made up of tubulin polymers. They are made up of tubulin protein. Microfilaments, also known as actin filaments, they are made up of actin, and intermediate filaments are made up of several proteins. Keratin is one such example. So talking about the structure here, you can see I have a picture. So I got all these pictures from the internet. So here we have microtubules. You can see the one, like a one tubule. Here you can see microtubules. So here these are hollow tubes, and the wall has 13 columns of tubulin molecules. So this is one, then two. So likewise, it has 13 columns of tubulin molecules, and it's a hollow tube made up of tubulin, and its main function is actually it's helping in the motility, in the movement. So when the chromosomes are needed to move in cell division, we know these microtubules come and then they kind of drag the chromosomes in opposite directions. So this is mainly by microtubules. We know that microtubules come and then they kind of connect with the centromeres and then they pull apart. So pull apart the chromosome. So this is helpful in chromosome movements and cell division, and also it's helpful in organelle movement. So movements kind of things are mainly by microtubules, and also maintenance of cell shape. So they maintain the cell shape as well. Microfilaments, actin filaments, they are made up of two actin strands. So here we can see these are microfilaments, and each microfilament is kind of made up of two actin strands, and each actin strand has actin molecules. So the main function is actually this is what where this particular filament forms the cleavage when the cells are dividing. We know the chromosomes are pulled apart, then the cell has to divide. So in the center cleavage is formed, and the cleavage is formed by this actin. So here you can see the tubulin. So this is how the tubulin comes, and here when the chromosomes are here, they pull it apart. So they reach the opposite poles of this cell. Then the one cell has to divide into two cells. You will learn about this in mitosis, which will come, which is in your book in the next following units. In the same unit you have several parts. In the second part you have this. It talks about the cell division. So there you'll understand microtubules connect to these chromosomes and they pull those chromosomes to opposite poles. So after they are pulled apart, here a cleavage forms so that these two cells are separated. It's formed by this microfilament, this actin. So this actin kind of here it forms a cleavage and it divides the cell into two. So here you can see cell division in animal cells, cleavage furrow formation, and also pseudopods. So the cell motility, it's also cell movement. So they form pseudopods. You all know about pseudopods. So for like amoeba, like it shows pseudopod activity. So for the cells to move, so unicellular organisms to move, these are helpful. So they form pseudopodia. And cytoplasmic streaming is another function. And maintenance of cell shape, again, as I said, all these help in maintaining the cell shape because that is what cytoskeleton does. And change in cell shape. In muscle contraction, you'll also be learning about that. In muscle contraction, actin, myosin filaments are participating. So actin filaments, the myosin filaments, the myosin head bind with actin and the actins are pulled. So it is useful in muscle contraction to contract the muscle. So that is what they have mentioned here. So these are the main function of actin filaments. And then we have intermediate filaments. So they are, you can see here, these are the intermediate filaments. So you can see that they are connecting, they are like a mesh, they are like a network, a mesh network, and they are connecting the whole cell. Between the two layers of cell membrane, you can find these intermediate filaments. One of several, so as mentioned earlier, different proteins are useful, and for example, keratin. These also maintain the cell shape, And apart from that, anchorage of nucleus, so they are anchoring the nucleus. As you all know, it's kind of between two cell membranes. These filaments are there, so they can anchor nucleus. Something in between these two layers, nucleus is between these two layers, so they can anchor the nucleus. And then formation of nuclear lamina, so it's useful in the formation of nuclear lamina. I hope you can remember which nuclear lamina is. As we all talked, here you can see this pink color layer. This is the nuclear lamina. So these intermediate filaments are useful in the formation of nuclear lamina as well. So these are the functions. Sorry. And now let's take a look at cilia and flagella. So these are actually extracellular parts, actually the continuation of the cell membrane, you can call it like that as well. So these both share a common structure. So they are made up of microtubules. You can remember microtubules. Earlier also we talked about them in cytoskeleton. So these are made up of microtubules with a 9+2 structure. So what is this 9+2? This is this 9+2. So in the middle we have central microtubules, 2 central microtubules, and surrounding it we have 9 doublets. You can see they are doublets. 9 microtubules, but they are paired, kind of 9 doublets. So this is the general structure for both cilia and flagella. Actually they are formed by basal body. The basal body continues as these cilia and flagella. So, but the basal body does not have this central microtubules. They just have 9+0 arrangement. So they do not have the central microtubules. They have 9+0 arrangement, and then when they come up, these 2 central microtubules are added. So to differentiate between cilia and flagella, cilia you can see here, cilia, these are cilia, these are tiny kind of, what do you call? These are very tiny tails, like tails, tiny parts. Here you can see flagella is very long. So that's one main difference. And also cilia are often organized in rows. You can see they are organized in rows. Cilia are numerous than flagella on the cell surface. Of course, here you can see only one flagella, but here you can see numerous cilia. So flagella, where you can find in human cell, is actually the sperm cells. The sperm cells have flagella, so they help in their movement. Cilia can be found in our respiratory epithelium. In our respiratory epithelium, it is actually ciliated pseudostratified columnar epithelium. So there you can find the cilia. So these cilia movements help in respiratory membrane. You all know we breathe, and when there are some foreign particles, they kind of help this cilia movement help to push them away so that they do not reach the lungs. So they are in our respiratory epithelium also. The cilia is helpful. The functions we can see act as a locomotor appendages, so in movements. So as I said, for sperm they are helping in movement, so act as locomotor appendages, can move fluid over the surface of the tissue. Of course, they help in movement of fluid over the tissue, especially the cilia helps for that. And then cilia lining in oviducts help move an egg towards the uterus. This is about female reproductive system. You all know there are eggs, and these eggs kind of they should enter this fallopian tube and they'll get into the uterus. So this, how do they get into this uterine tube is via this cilia movement. So these cilia move and they help to move the egg towards the uterus. So these are the 3 functions. Finally, we have centrioles. Centrioles are also somewhat related to Cilia Flagella because these also have this microtubule arrangement, but 9 plus 0 arrangement. Outside we can find 9 triplets. So there are 3 microtubules. You can see 1, 2, 3 and 9. Altogether there are 9 but arranged as 3. So 9 triplets. We can call it as 9 triplets. 3, 3, 3. But in the center we don't have any central microtubules. So 9 plus 0 arrangement. So here you can see each centriole is composed of 9 sets of triplet microtubules arranged in a ring. So these centrioles are very helpful in cell division. So these centrioles are moving towards the opposite poles and then they kind of form these spindle fibers, and these spindle fibers attach to the chromosomes and they help to divide the chromosomes into opposite poles. So they produce aster and spindle in cell division. So this is its function. So I hope you understood what I talked about today. So this is how I am planning to continue the rest of the units in your GCE AL syllabus, in your curriculum. So hope today was also today's this one also helpful for you all. Please do mention how it was. If I can improve in any way, please do mention that as well. I'll definitely reply to your messages. So thank you so much for listening. Please do share this with your friends, subscribe the channel, like the video and tell me how I can improve. Bye.